Thank you again, DDA, for inviting me to give this talk on insulin, on the signaling cascade for energy supply in exercising muscle from normal and insulin resistant subjects. And when we talk energy supply in exercising muscle, we typically talk about glucose uptake, which is supplying the exercising and energy demanding skeletal muscle. And that's of course very interesting, but what when I thought about this, what I think is even more interesting, and maybe in relation to the prevention and treatment of at least type 2 diabetes, is to look at energy supply in the post-exercising skeletal muscle, because this is maybe quite relevant when we talk about insulin resistance and reversal of insulin resistance in various subjects, including type 2 diabetes. So the main part of my talk will actually revolve around energy supply, or at least when we talk about glucose uptake in the post-exercising skeletal muscle. I'll also talk a bit about energy supply in exercising skeletal muscle. So in light of this, the outline of my presentation will include a brief, you could say, historic overview on exercise and its potential to prevent diabetes, type 2 diabetes. Then I'll talk about exercise training and acute exercise, and how these two exercise modalities affect insulin action in muscle, and I'll talk about the mechanisms responsible for these effects. So before I'll move on, I'll like to present to you the take-home messages. And the first take-home message is that it's actually not that new that we know that exercise affect insulin action. We've known that, or we have had you can say the population, the community has been knowing this for quite some years now. Then metabolism during exercise, that doesn't seem to be compromised in insulin resistant skeletal muscle. That's of course important, but maybe not that interesting. And then third take-home message is that exercise training affects insulin action, but it's likely not a cure for type 2 diabetes. It enhances insulin action, and not only exercise training, but also a single exercise bout enhances the action of insulin in insulin-resistant skeletal muscle. And then the whole body insulin action, which may be most relevant for most people, it depends both on acute and chronic effects, both on the acute and chronic effects of exercise. So for the historic overview, already actually in the ancient time, described observations from various physicians indicated, you could say, a positive effect on exercise in the prevention of diabetes. And many of them actually noted that people with increased urination, or also known as polyuria, and that is typically characterized by sweet urine, could benefit from exercise, thereby decreasing the need to urinate. This, at this time, diabetes per se was not diagnosed, but actually the Greek physician Aristeas actually coined the term diabetes, who discovered that people with diabetes has this sweet urine. So in the early therapeutic period before insulin was discovered, various diabetologists also noted and recommended exercise for the treatment of diabetes, and it actually became say worldwide accepted that besides dietary interventions, exercise was one of the ways to reverse the symptoms associated with diabetes. Then in the late therapeutic period, so you would say after the discovery of insulin by Banding and Best in Toronto in 1921, it really quickly became apparent that insulin exerted enhanced effects when combined with exercise, that in the end was described by increased exercise-mediated capillary flow and thus enhanced delivery of insulin to the muscles. And then especially due to the work of L.A. Joslin, who was the first US medical doctor to really specialize in diabetes. And he more or less became a world spokesman for the cause of diabetes. Well, he added that exercise in addition to insulin and diet was included as one of the cornerstones in the treatment of hyperglycemia and diabetes. All right, so exercise is good for diabetes. However, when looking at how exercise and insulin function to improve hyperglycemia or diabetes, it is quite interesting to note that exercise and insulin actually work by similar ways to reduce blood glucose levels, and thereby you could say reverse symptoms of type 2 diabetes. So in this, you could say, seminal paper by Sisman and colleagues, it was shown that insulin and exercise in the form of contraction increased glucose uptake in isolated skeletal muscle from wild type mice, but that this effect was blunted in skeletal muscle from mice lacking the GLUT4, glucose transporter GLUT4. However, around the same time, it was also reported that exercise increased glucose uptake in muscle by insulin-independent mechanisms. As exercise, you can see here, increased glucose uptake normally in muscle lagging the insulin receptor. So exercise increased glucose uptake via GLUT4, but independent of the insulin receptor. And this notion is really also supported by the, you could say, pioneering studies of John Warren, who reported that the glucose clearance during exercise was at least as great in type 2 diabetics as in controlled subjects. And this kind of these observations kind of led to the general notion that insulin-resistant subjects exhibits normal exercise regulated metabolism in muscle. And that's actually both in regards to glucose and fat metabolism. So that's nice and important. Yes. So again, the take-home messages are that the effect of exercise on insulin action is not new. We have known that for many years. And metabolism during exercise, that's not compromised in insulin-resistant muscle. And actually, if hyperglycemia is present, which is typically the case in patients with type 2 diabetes, muscle glucose uptake and glucose oxidation may actually be elevated because there's a higher gradient and therefore you have higher glucose uptake. So metabolism during exercise is not compromised. So that's good. And that's important in these patients. But you could say, considering the amount of time spent doing actual exercise during a day, it may not be what is going on in the muscle during exercise, but rather you could say after exercise that benefits patients with type 2 diabetes in the long run. And as you probably have heard earlier today, skeletal muscle is the main thing for glucose disposal during insulin stimulation. And since muscle insulin resistance is a major defect in type 2 diabetes, finding ways to reverse this defect may, you could say, constitute a treatment strategy for type 2 diabetes. As you may have guessed, exercise may also constitute such treatment strategy, as exercise enhances insulin-stimulated glucose uptake in skeletal muscle, as I also noted in the outline. So the first real evidence that exercise could improve insulin action or insulin-stimulated glucose uptake in muscle were provided by Bjørn Top and colleagues in 1972, and he noted that lifelong physical active men had better glucose tolerance a couple of days after the last exercise bout, even when plasma insulin levels were lower. So this really suggests that exercise has this potentiating effect on insulin action in muscle. And then later on, a couple of years later, at a Kroc symposium in 1978 in the US, it's a symposium on diabetes and exercise. Although it was only published as preliminary results, it was reported that months of exercise training improved oral glucose tolerance in non-obese middle-aged men with chemical diabetes. And also by a physician, Neil Ruderman, he also reported that glucose disappearance rate during an intravenous glucose tolerance test increased in impaired glucose tolerance, in men with impaired glucose tolerance after 6 weeks of exercise training. But he also noted that this effect actually reversed even 14 days after they stopped training. So they showed that exercise could increase insulin action, but that it also quickly reverses if you stop exercise. All right, so looking at how insulin action in muscle adapts to exercise training, we now know that exercise training, at least in many cases, increases muscle mass. That, of course, will increase the overall amount of glucose taken up by muscle in response to a stimulus, insulin stimulus. Furthermore, exercise training increases capillary density, and it also increases insulin, calorie recruitment by insulin, which improve, you could say, glucose delivery to the muscle. So the muscle are able to take up more, at least the muscle are handed more glucose to take up. And then exercise training also improves muscle protein quality. We know that exercise training enhances the membrane permeability. We know the muscle receives more GLUT4. We know that exercise training increases mitochondrial density and a lot of glucose handling proteins, which improves both the extraction of glucose from the blood, but also improve metabolism of glucose in skeletal muscle. All right, so then 10 years ago, an attempt to illuminate how exercise training affects diabetic muscle at the molecular level, the lab of Kurt Højlund and Jørgen Wojciechowski performed a 10-week exercise training program in type 2 diabetic patients and matched control subjects. And they showed that the glucose disposal rate increased following 10 weeks of exercise training. But as you also may notice, this increase in glucose disposal rate following exercise training were not rescued in the type 2 diabetic patients compared to the matched control subjects. So they actually didn't even reach the before measurement in the control group. So this suggests that exercise improves insulin action in muscle, but it may not be an actual cure for type 2 diabetes. So So when they looked at various glucose handling proteins in the skeletal muscle, and this is a simplified cartoon on how insulin, and you probably have seen this, how insulin affects or increased glucose uptake in muscle. The author showed that many of these proteins that are involved in insulin-stimulated muscle glucose uptake, the expression of these proteins increased with exercise training, but also acute insulin signaling, such as phosphorylation of this protein, now known as TBC1D4, was rescued by training, all of which could explain improvements in glucose disposal rates in type 2 diabetic patients after exercise training. So yeah, from this we know, and I showed you an example, but we know that exercise training enhances insulin action in muscle. It relates to enhanced expression of effector proteins. It could be more GLUT4, it could be more hexokinase, which is a protein that phosphorylates glucose when it enters the muscle cell. Some signaling defects, such as defects on TBC1D4 and phosphorylation defects on TBC1D4, were rescued by the exercise training in type 2 diabetic patients. But also, as I mentioned, training is maybe not, this of course was only 10 weeks, but this study suggests that training may not be a cure for type 2 diabetic patients, as they don't really reach the same level as matched control subjects. All right, so when we turn away from exercise training and look at how acute exercise modulates insulin action, and it's actually been known for almost 40 years now that acute exercise is also able to affect insulin action. So this is just a single bout of exercise that subsequently, a couple of hours after, increases insulin action on glucose uptake in skeletal muscle. And so this is a small cartoon showing this, and we know during exercise, as I showed you, glucose uptake increases in muscle, and then when we rest, subsequently rest, glucose uptake decreases to resting levels in the prior exercise muscles. But then when muscles are then stimulated with insulin, for example during a meal, glucose uptake increases to a further extent in the prior exercise muscle compared to the prior rested muscle. So this we have known for many years, and I'm heavily involved in looking at which mechanisms in muscle are responsible for mediating this effect. So the physiological role of this is to partitionate carbohydrates toward the muscles in need, because when you exercise, you decrease your glycogen stores, and the muscle would like to rebuild these stores, and it does this by enhancing insulin sensitivity, which increases glucose uptake and then increases the ability of muscle to resynthesize glycogen. And we also believe that this effect of enhanced insulin sensitivity is important for muscle glycogen supercompensation, which we believe could be important when we perform exercise, at least aerobic endurance exercise. This effect we have observed in many species, which therefore we believe is important since it's conserved. It's been observed in various insulin-resistant states. We observe it in isolated muscle, but also in the in vivo setup, so in humans. But also, if you take out a muscle and then stimulate it to contract, we can also see enhanced insulin sensitivity, suggesting that this insulin sensitizing effect of acute exercise is mediated by local factors within the muscle. And then it depends on the exercise type and duration, and it also depends on carbohydrate availability. So if you eat a lot of carbohydrates, this effect of exercise and insulin action reverses faster. Right. So a single exercise bout enhances the action of insulin in muscle, and it's been reported both in healthy and insulin-resistant subjects. Then what are the mechanisms? Well, we believe there are at least two mechanisms that are important. We believe that this enhanced insulin sensitivity after acute exercise depends on enhanced capillary recruitment by insulin, which improves delivery of glucose to the muscle, and then enhanced intracellular signaling events, which improve extraction. So enhanced delivery, so more glucose to the muscle, and then the muscle are then able to extract this enhanced or this improved delivery. So yeah, so in 2017, we provided the first evidence in humans that enhanced capillary recruitment by insulin is necessary to drive enhanced insulin-stimulated glucose uptake after exercise. And we showed that microvascular muscle blood flow, this is 4 hours after exercise, was nearly similar between the prior rested and prior exercised muscle. However, in response to insulin, microvascular perfusion increased in the prior exercised level to a greater extent than the prior rested muscle, which coincided with this increase in leg glucose uptake in the prior exercised muscle compared to the rested muscle. And then when we added a nitrate oxide inhibitor, which decreased microvascular flow in the muscle, we also noticed a decrease in this enhanced insulin-stimulated leg glucose uptake. And then we removed the inhibitor, glucose uptake rose again higher in the prior exercise leg. So suggesting that that exercise needs, or prior exercise increase the effect of insulin to increase microvascular flow. So which means that the muscle are able to extract more glucose from the circulation. So a single exercise bout enhances the action of insulin in muscle, and it's dependent on adequate delivery of glucose, which is mediated by improved capillary recruitment by insulin. Then what's been known for many years now is that the insulin action after exercise, so improved insulin action on glucose uptake, is fairly dependent on, or we think it's fairly dependent on GLUT4 in the plasma membrane. So, and I won't go too much into this, but in this study, they looked at glucose transport in prior exercised rat muscle, and you can see here in insulin-stimulated prior exercised muscle from rats, glucose uptake is higher compared to the rested, and that occurs concomitant with increased amount of GLUT4 in the cell surface membrane. So enhanced insulin sensitivity after acute exercise associates with GLUT4, with more GLUT4 in the plasma membrane, not more synthesis of GLUT4, but the pool we have, or the pool muscle has, is more of this is recruited to the plasma membrane. Yeah, so to make a long story short, we will investigate a lot of proteins, and these proteins in the insulin signaling cascade, and these proteins does not seem to be regulating the enhanced effect of insulin after exercise. But then some 10 years ago, it was shown that enhanced phosphorylation of TBC1D4 coincided with enhanced insulin sensitivity after acute exercise. At that time, it couldn't really be explained how enhanced phosphorylation of TBC1D4 could occur without enhanced upstream signaling up here. But then it was discovered that the exercise-activated protein kinase, AMPK, also targets TBC1D4, which made TBC1D4, you could say, a kind of convergence point for insulin and exercise signaling that likely could explain this enhanced insulin-stimulated glucose uptake after acute exercise. So in light of this, actually I did a couple of experiments where I wanted to test whether AMPK was involved in regulating enhanced insulin sensitivity after acute exercise. And we anesthetized some mice, and then we, and these are, you can see over here, these are breathing alive mice that are anesthetized, but then we hooked on some electrodes and then stimulated the muscles of the lower hind leg to contract. And after this, we took out the muscle, we had them recovery, we had them in a tissue bath for 3 hours without insulin, and they recovered there for 3 hours, and then we stimulated the muscle for 30 minutes with or without insulin. So this is a highly controlled situation where we try to mimic what is happening with the exercise and insulin sensitivity afterwards. And what we showed, or what I saw, was that in wild-type mice, during a sub-maximal insulin concentration, insulin-stimulated glucose uptake were higher in the prior contracted muscle compared to the prior rested muscle. But in my AMPK knockout animals, this effect was gone, and there was no effect with the highest dose of insulin. So this really suggests that AMPK is necessary to induce enhanced insulin sensitivity after a single bout of exercise. And when we look downstream of AMPK and looked at phosphorylation of TBC1D4, we also noted that in the wild-type mice, in the prior contracted muscle from the wild-type mice during insulin stimulation, phosphorylation of 2 sites on TBC1D4 were markedly enhanced compared to the rested muscle. But in the knockout animals, there was an effect of insulin, but there was no enhanced signaling by prior exercise. So enhanced glucose uptake by insulin coincided with enhanced phosphorylation of TBC1D4 in the wild-type mice. So this led to the model that we believe when we do exercise, we activate AMPK, that's phosphorylates TBC1D4, it kind of modulates it, it primes it. So when insulin binds to the insulin receptor some hours after, we have hyperphosphorylation of TBC1D4, which liberates more GLUT4 vesicles to fuse with the plasma membrane, thereby increasing glucose uptake in skeletal muscle. So a single bout of exercise enhances the action of insulin in muscle. It's dependent on GLUT4 translocation. It's not dependent on increased protein expression. It occurs independent of altered proximal insulin signaling, but it's dependent, we believe, on AMP kinase. And I haven't shown you the data, but we have also done the same experiments in TBC1D4 knockout mice, and we see the same in these mice. So TBC1D4 is also important. So in light of this, well, you can say in light of this, or in summary, we now know that insulin sensitivity, or the insulin sensitivity index, you could say, in healthy and insulin resistant muscle increases during a period of exercise training. It increases every time we do a single bout of exercise, but it also increases in response to the combined effect of each single bout of exercise. So the effect of training is marked by this red line, and the effect of a single bout of exercise is marked by this black line. And the effect of acute exercise and exercise training on insulin sensitivity is occurring by totally different mechanisms. So in the end, this means that whole body insulin action depends, again, both on the acute and chronic effects of exercise, and that patients with type 2 diabetes benefits from exercise training to a similar extent than as their match control subjects. So it may maybe it went a bit fast, but I think my time is up, and of course we'll take happy to take any questions you may have.
Let's see if we have any questions yet. Not yet. Okay, then I will give you a question, Rasmus. it's very interesting with the increased insulin sensitivity, and I was wondering whether, you know, type 2 diabetes has appeared to be complex, as some diabetics are insulin resistant, some have reduced insulin production from the beta cells, etc. Is there a difference in response in insulin sensitivity in these different types of type 2 diabetic patients? Do you know about that?
No, we don't actually know, and to be honest, there's no one who's actually done the correct experiment to test that muscle from type 2 diabetic patients are also enhanced by an acute bout of exercise. We know from insulin resistance, so you could say pre-diabetic patients, that they also enhance muscle insulin sensitivity after an acute bout of exercise, but then we have only whole body measurements to suggest that type 2 diabetics also benefit from an acute bout of exercise. Having said that, I strongly believe that type 2 diabetics also benefit from this, and I think it's only a matter of doing the experiment and showing that they respond normally, or as well as controlled subjects. Regarding whether, so type 2 diabetes is a multifaceted disease, and I haven't a clear answer, but I would my gut feeling says that they will benefit equally well, whether they have, depending on where the disease is located in type 2 diabetic subjects. So no matter whether it's the pancreas or the liver or the muscle, I think they will all benefit from a single bout of exercise. At least there's no data to suggest otherwise out there, so in animal models and so on. So I believe that there's everybody will benefit from this from an acute bout of exercise.
Yes. Thank you. And now we have a lot of questions. The first one is from Victoria Asgaard Mikkelsen. She writes, "Hi Rasmus, thank you for a very interesting talk. Could you elaborate on why different types of exercise have different effects on muscle insulin sensitivity? Is it the same mechanism, and which types of exercise are more effective as insulin sensitizers?"
Yeah, so you could say we typically categorize exercise training into two. So we have resistance exercise, and then we have aerobic endurance exercise. And of course, when you do resistance exercise, you increase your muscle mass to a greater extent than if you do endurance types of exercise. And just by increasing your muscle mass, you will also increases the ability of insulin to reduce blood glucose levels. So just by doing this, you'll gain an effect. But you'll also but if you look at the muscle per se and take out the size or the bulk muscle mass, then you'll get most out of it if you do endurance types of exercise, which we know will increase as a road the capillary density of the muscle, so the muscle are able to extract more glucose. It will increase these glucose handling proteins in muscle to a greater extent than resistance exercise. And you'll also you'll get more mitochondria, so you'll increase metabolism, or you'll increase the capacity for metabolizing glucose in muscle. So if you want to have a very sensitive muscle, and at least in regards to insulin and glucose uptake, endurance types of exercises is to prefer. If you look at an acute bout of exercise or endurance types of exercise and resistance exercise, not much research has been performed on resistance exercise, and that has been performed shows no effect of acute resistance exercise on insulin sensitivity. So if you go down and pull some weights, you won't increase your insulin sensitivity acutely in skeletal muscle. But there's not a lot of research on this. But the research present don't suggest at least a enhancing effect of resistance exercise, whereas we see this with acute exercise. And then I had a question when I was in the breakout room. And the question was more or less, how low can you go? So the intensity of the endurance exercise. So how intense do you need to do your exercise before you receive this enhancing effect of exercise? And to be honest, we don't know, but we have performed an experiment where we do a low or 70% of maximal exercise capacity, and then 70 and 95% of maximal capacity, and when we see a similar effect on the acute enhancing effect on insulin sensitivity of the exercise. So it suggests that this effect of acute exercise on insulin sensitivity is a switch on, switch off mechanism. So you need to go above a threshold, and then you kind of activate it. How low you can go, we don't know. But you probably need to go above 50 to 60% of your maximal capacity, because that is where we believe we activate AMPK. That's where we see activity of AMPK, which we believe is important for this phenomenon. So at least 50 to 60%, I believe you need to do to get this enhancing effect of exercise. So you don't get it if you go down the stairs or take a walk, a slow walk down the street.
You have to sweat. Okay. Yes. And the next question is from Pernille Hellman. Hi Rasmus, thank you for a very nice, interesting talk. Could you elaborate?
It's the same question as asked before from Victoria.
Okay. Yeah, I see. Okay. Yeah. Good. Then thank you. Then Maria Winter Arcu says, nice talk. Was there any phenotype of the TBC1 D4 KO knockout mouse where other tissues than muscle affected?
Well, so this study I performed, I actually studied in a muscle-specific TBC1 D4 knockout animal, and we are currently characterizing the model, but it has a fairly mild phenotype, but so there's not that much we have discovered other than that TBC1 D4 in muscles seems to be important for this enhancing effect of acute in this case acute contraction of the lower hind limb. We are still investigating whether a lack of TBC1 D4 in muscle also affects whole body insulin sensitivity after the mice have been running on a treadmill. Yeah, so if that was, I guess, hopefully that was an answer.
Thank you. And Jule Brøsen asks, yeah, it's kind of the same answer, but it's a question, but the last line maybe, how do we transfer the, sorry Rasmus, did you see the question? She asked what is necessary in terms of exercise. Is physical activity enough, or does it need to be strenuous exercise? You answered that, but then she asked, how do we transfer this to the clinic, and what should we expect from the patients? Like, you could maybe elaborate a bit on that.
Yeah. So how do we transfer this to the clinic? Yeah, well, I typically when people ask me what to do, and then I typically respond to them whatever it takes to get your ASS out of the couch, right? So a little is better than nothing, and if you can't perform high intensity exercise to increase your insulin sensitivity, then just do the walk, because then you'll also get a lot of beneficial effects from walking. At least if you do, you could say, if you do, there's data suggesting that if you do interval walking, for example, you'll actually get some quite beneficial effects. So that's how to implement it. You know, whatever it takes to get you do some kind of training, because you will get better, and I would say no matter how little you do, as long as you do something. Yeah, so that's my best advice. Yeah.
Yes, less is more, exactly. And Annemette Freubert asks, are there any studies that investigates if AMPK can be phosphorylated therapeutically,
Yeah.
thereby mimic the actions of exercise and increase insulin action?
Yes. So that's a good question, and so I think you're thinking about the exercise pill, right? So instead of exercising, could we then leave our ASS in the couch and then take a pill and then have the same beneficial effects as performing 1 hour of running? And the short answer is yes. There seems to be some evidence to suggest that it's possible. I've been involved in a huge study, collaboration with the Pfizer medical company in the US, where we actually developed a specific AMPK activator, and this activator was provided to mice and non-human primates, and we showed that mice, diet-induced obese mice, if they receive this AMPK activator, their insulin sensitivity improved, glucose uptake in muscle improved, mitochondrial density in muscle improved. So overall, their metabolism improved, and also in the monkeys, when they received this compound, their metabolism, insulin or glucose metabolism improved. So the short answer is yes. Whether it works in humans, we don't know. I believe it would be. There's of course some concerns when applying these drugs, and one concern, at least for AMPK activating drugs, is that if you activate AMPK in the heart, you typically see glycogen accumulation, and you don't want glycogen to accumulate in your heart, because that could cause some severe heart damages. So there are some concerns using these exercise pills, but if you could take a pill and then direct it towards the muscle, so you only affect the muscle or affect proteins that are only expressed in muscle, then of course I think that's a huge potential for exercise pills. And then you could say, there's ethics concerning this, and who do you want to give this pill to? Do you want to give it to obese, lazy subjects, or people who compete in various sports? And that's I guess that's not my I guess I'm not the right one to answer that, but at least we see some potential in giving these exercise mimetics or exercise pills to patients that are, for example, hospitalized for various reasons, or have various handicaps because they have been in an accident. So it's not it's not only bad things that comes from these exercise pills. We believe that there's a true potential, and we can various peoples can benefit from this.
Thank you, Rasmus. And we don't have any more time, but we have 2 questions left.
So I'll make it short. Yes.
Perfect. So the next question is from Mette Glavin. And I can see the rest of the names, but they are asking, is there any indication that the chronic effect of exercise keep making insulin sensitivity better, or is there a maximum level?
Yeah. So I also talked about this in the breakout room, and there seems to be like an upper level. From the literature, highly trained individuals have sky high insulin sensitivity in muscle, and then it seems that as if they don't benefit as much as non-trained people, at least with the acute bout of exercise. So they have reached a ceiling and they don't benefit from this acute bout of exercise. It looks like that. Sorry.
The maximum level, yeah. Okay. And the last question is from Felix Bo Pedersen. Is there any type of exercise that seems to be better than other? And are there any kind of memories? So after training for a longer period and then detraining, can you then faster activate better insulin or achieve better insulin sensitivity when starting to train yet again?
Yeah, so that's maybe not my field of expertise, but yeah, there may be some indication, and that's maybe on a DNA level, that if you have been trained, then the muscle remember being trained, so it gains faster the insulin sensitivity than it had when it was trained. So there seems to be some data suggesting that muscle has memory. So when you have been at a certain level, you'll get to that level faster when you start training again. And you also see that especially within resistance exercise and muscle building, that if you have been you could say huge before, then you'll get huge faster again when you start doing resistance exercise. So and there may also be some data indicating that that's what is going on with endurance type of exercise and insulin sensitivity. But that's not my field of expertise.